So I am, as all of you know, my name is Amar Basu, and I'm in the electrical engineering department. So I'm going to actually start today with the shameless plug. Since I didn't really prepare a slide for this, I'm just going to go to our lab's website. Our lab works on microfluidics. So we're, like Professor Lam said, we are right across the street here in the electrical engineering department. A lot of our work focuses on device design. This is an example of a microfluidic chip that we recently designed. It's designed to store 20,000 partitions, 20,000 droplets for digital assays. The digital assays is going to be the subject of my talk today. Our two general areas of research includes multi-phase microfluidics for high throughput studies. So many of you are in this area, in the biotech area. So you probably have an idea that now biotech is moving towards large-scale screening, things like genomics, proteomics, where you need to do a lot of experiments. And when you're doing a lot of experiments, it's often beneficial to miniaturize those experiments so you don't consume as many reagents and so that your reactions happen faster. So what we do is we miniaturize a lot of like these high-throughput type of assays that are typically done in the biotech industry these days, and we make the instruments to do them. And so, we are an engineering lab, so we like building stuff. We use microfabrication, nanofabrication. We do a lot of simulations. We build a lot of devices for the biotech field. That's one of our areas. The other area that we work on is wearable and portable electronics for health and environmental monitoring. So if you're interested in working in wearable sensors or small devices that can monitor water quality, if you have a project idea that you'd like to explore, come talk to me, email me. I have office hours twice a week. We can chat about it. I can bring you and give you a tour of the lab if anyone is interested. So that's my shameless plug for my lab. If any of you are interested in working in this area, I'm also I'm a little bit sick today, so I might be sitting and standing both as we do this presentation. So some of you may know me before because I have given seminars at BME before. So in the past, I've given seminars just about the research projects that I've done in the past. I thought I'd change it up a little bit this time and talk to you about an emerging area of biotech, which our lab has started to work on a few projects on. But I actually spent 6 months last year writing a review paper on this topic, so I thought I'd share this area with you. And one of the reasons why I think you'll find this interesting is because this is actually one of the hottest areas of biotech right now, this area of digital assays. How many of you are working in doing research in the biotech area where you're, you're working with cells, working with proteins, DNA, or you're trying to detect something, detect chemicals? Okay, show of hands. 1, 2, 3. Okay, so what are you all? What are the types of projects you all are working on right now? Please go ahead.
We're both working Raman spectroscopy, immune stuff, so virus, bacteria, T-cell.
Detecting viruses and bacteria.
Yeah.
Great.
Pathogen detection.
Pathogen detection. Excellent. Excellent. You'll see. I think you'll find this quite interesting then. And in the back.
I'm trying to detect a kind of cell in the CSF, so finding out proteins and stuff like that.
You're trying to detect cells in the cerebrospinal fluid?
Yeah.
Okay. Yeah. So digital assays is actually a nice tool for detecting low amounts of proteins and low amounts of DNA in biological fluids. I'll talk about that. And I think in the back, you would also raise your hand.
A glucose O2 biofuel cell.
I'm sorry?
I'm building a glucose O2 biofuel cell.
Glucose biofuel cell.
Yeah.
Ah, very interesting. Okay, I don't know if digitization would help that type of assay, but we should talk afterwards. I think there are some on this side of the class. No? Okay. So we'll keep the format conversational. So if at any point in this presentation I'll ask you questions, or if you have any questions, just feel free to either raise your hand or just blurt out your question. No problem. We can keep it more like a conversation. In fact, if we do it that way, it's better because sometimes I can tend to go on about things not realizing that maybe the audience missed an important point. Okay. So I'd rather you learn something about a small portion of it than to get the entire presentation but not know it very well. Okay. All right, so here's the stuff we're going to talk about. Analog versus digital assays. We'll talk about the mathematics of digital assays, how you would implement them, and then we'll talk about 3 types of digital assays. And the 3 types of digital assays correspond to the 3 types of biomolecules, bioparticles. So we'll talk about DNA. So digital PCR is a way of detecting DNA with very, very high sensitivity. Digital protein assays, a similar way to detect proteins in biological samples with very, very low limits of detection. And digital cell assays, and some of you have also heard this term, single cell assays. Single cell assays allow you to investigate biology at the single cell level and measure the properties of single cells. And you could do that with very, very high throughput, meaning you could measure the properties of thousands of cells in a single experiment. And that what that allows you to do is it allows you to look at differences between cells. All right? And that's one of the big areas that folks are interested right now. The research community is very interested in right now. And we're probably not going to have time to get to the work that I do in my lab. So what I'll do is if any of you are interested in this area or interested in the topics I mentioned earlier, just come email me and come talk to me. We can arrange a meeting. And I will also be teaching, I'm supposed to be teaching the biomedical microsystems class. It's a 7,000 level course next semester. Okay, so I like to start at the basic level. What is an assay? Just so we're all on the same page. Some of you may know this already, so bear with me. A quantitative assay is a test where we want to measure the amount of analyte in the system. So we want to say there are 10 nanograms of this particular protein in the sample. A biological assay could look something like this. This is the NIH High Throughput Screening Center. This is where they do the assays on these micro well plates. So they're doing hundreds or thousands of assays in succession. And they're usually trying to test some kind of drug compound against a cell of interest. Another type of point of care assay is something like this. If you've done Wellness Warriors, you've seen this device. This is the Cholestech cholesterol analyzer. If you go to those wellness screenings, they'll poke you with a needle, and they'll take a drop of your blood and put it into this small chip that you see here. And this chip will actually separate out your blood components into the hematocrit and the serum. It'll analyze the serum to look for the amount of HDLs and LDLs and cholesterol that you have in your blood, and in like a minute or two, it'll give you a reading. All right, so that's an example of an assay. We're trying to measure the specific quantity of a specific target analyte. And so the general schematic of what that would look like is something like this. You start with the sample, all right, and that sample goes into some type of measurement device, and the typical readout mechanisms for assays include spectrophotometry, fluorescence, and electrical conductivity. So these, I'll give you a few examples of these, but these readouts are related to the sample concentration, and you usually will make some type of a calibration curve where you'll take various concentrations of your sample and you'll plot the signal against the sample concentration. So this gives you a calibration curve. So when you then look at an unknown sample, you can use this curve to figure out, if you're getting a signal somewhere here, then you can look up on this chart, look up on this curve what your sample concentration is. So the important thing about a traditional assay is that the concentration is proportional or some ways related to the analog signal that's given by your readout device. So if we take a few examples, probably the most common type of assay are different types of optical assays, right? It's just so we're all on the same page here. Molecules absorb light. Okay, proteins absorb light. DNA absorbs light. So one way of detecting DNA is to you have a light source here, okay, and that light source goes through something called a monochromator. Monochromator selects a specific wavelength. Okay, DNA typically in the UV range. You'd want to use the UV range for DNA. For proteins, also you can use ultraviolet. For detecting hemoglobin in blood, you want to use something like 700 nanometers or in that range. All right, so you'll select a certain wavelength depending on what you're trying to detect. And here's the important part. The light goes through the solution, and then it goes into a detector. So in the case of spectrophotometry, the signal that you're getting is proportional to the concentration of the analyte. And this is Beer's law. Excuse me. I'm going to grab a cough drop. So the more of the molecule you have in the solution, the more it absorbs, right? So it's given by Beer's law. The transmitted light over the incident light is equal to e to the negative eta times Cl. So notice that this C is a concentration. Okay, so based on the light that's detected, you can back out what the concentration is. Fluorimetry is another technique. This is where you would label the sample first with something called a fluorophore, and fluorimetry is fairly straightforward. The amount of light emitted by the sample, in this case, fluorescence excites a sample. So you excite it with blue light, and it'll give off green light. That's one example. So you'll detect the amount of green light being given off, and equate the amount of fluorescence emission to the concentration of the sample. All right. So the question is then, what is a digital assay? Let me just highlight the fact that in both of these cases, the higher your sample concentration was, your signal goes up in some type of monotonic manner. All right. So you're trying to equate the concentration to an analog signal. Now, for those of you who've built systems, you know that has some inherent weaknesses associated with it, right? If you have a bad detector, then your signal will be noisy, and that's going to reduce your limit of detection or make your limit of detection worse. You won't be able to detect small concentrations. The other issue that often happens is these curves are not always linear. As scientists, we like linear curves, right? Like if you double the sample concentration, you'll double the signal that you get from your spectrophotometer. Now, in a lot of cases, this is true, but in spectrophotometry, for example, you don't always get a linear curve. Once you start getting to high concentrations, it starts to tail off. If you don't have a linear response, you have to make a calibration curve. And trying to make a calibration curve when you have very little amounts of sample, let's say you have a patient sample, and you're trying to make a calibration curve out of different concentrations of an analyte, it's very difficult to do when you have very limited amounts of sample. So let's look at what the concept of a digital assay is. All right. In a digital assay, you're essentially counting molecules. Instead of looking at sample concentration, you're actually counting individual molecules. All right. This slide compares an analog assay with a digital assay. So in an analog assay, you do this bulk measurement. In a digital assay, you do this step called partitioning. This is where microfluidics comes in. You take the sample, let's say the sample was 1 microliter or something like that, and you partition it into many, 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 many small containers. We're talking like 20,000, 100,000, maybe even up to 1 million. And there are microfluidic systems that enable you to do that. So why do you want to partition? Well, back in the 90s, Kurt Vogel from, I believe it was Duke, he came up with this concept of a digital assay. If there are more partitions, if you break your sample up into partitions, and if there are more partitions than there are molecules in the sample, what's going to happen? You're going to actually end up with some partitions that are empty and some that have molecules in it. Do you see this? This is an important point of digital assays. I'm going to zoom in on this here. So let's say the sample you're trying to detect is DNA. All right, we talked about some, what was your name?
Kayla.
Kayla and Caitlin, okay. So Kayla and Caitlin are looking for low concentrations of viral DNA. Was that fair to say, or bacterial DNA? That's one way to do detection?
Yes.
Okay, so let's say you're trying to detect DNA in your sample, right? The way the digital assay would work is that you would first partition it into a gajillion small containers. Enough containers, there's more containers than there are molecules in your sample. And what happens then is that some of your containers are going to have maybe one nucleic acid, maybe two, just a very few number of molecules in each partition, and other partitions are going to be completely empty. It turns out that if you're in this zone, if there are typically less than 5 molecules per partition, you can use Poisson statistics to actually figure out the actual number of molecules that you had in your sample. So instead of measuring a concentration like so many nanograms per milliliter, you are actually saying, I actually have 1,536 DNA sequences corresponding to this virus. I mean, it's a whole new level of precision, right? If you can actually count individual molecules. So that's what digital assays are all about. So you'll take a digital measurement in each partition. So you have to look at every single partition and determine whether there was 0 molecules in there or whether there was 1 or more molecules in there. And so your signal is going to look something like this. And in each one of the partitions, you're either going to have a 0 or a 1. 0 means it was empty. 1 means there was 1 or more molecules inside that partition. And after you do that, you can use Poisson statistics to actually figure out what the number of molecules in the sample. And Poisson statistics actually uses the number of empty partitions. The concentration is proportional to, actually is equal to the negative log of E. E is the proportion of empty partitions. So let's say 35.6% of them were empty. Then you know what the concentration is. All right. So, yes, please go ahead.
So, so this assumes that the number of DNA samples in a full partition is roughly the same. How do you account for, is there ever a time where one will have 1 and another will have 20 samples or DNA sequences in it?
Yes. So what you're bringing up is something called subsampling error. So subsampling error turns out is a problem not just with digital assays but with all assays. If I have several liters of blood in my blood vessels, right? If I just take out a 20 microliter sample of blood, well, is it really representative of the my entire blood, right? It turns out the mathematics for that are very well established. It's the standard deviation is equal to the square root of m, where m is the number of molecules that you expect to detect. So if you're detecting a lot of molecules, not a problem. If you're detecting very few, like less than 100, then this can become an issue. Excellent question. So why do digital assays? So the first thing is this last point here. No calibration curve needed. So we just talked about that, right? We're counting individual molecules, so there's no calibration curve in this sense. There's 2 other effects also which turn out to be very useful. This partitioning does 2 very interesting things. Have any of you dealt with inhibitory effects in an assay? Does anyone know what inhibition is? Anyone? Don't be shy. Yes.
So really the prevention of an action taking place?
Prevention of an action, right. So in the context of an assay, inhibition is when, let's say you're trying to detect a nucleotide of this virus, right? The virus has a certain sequence. There's a very small amount of that viral DNA, but there's all this other DNA in the solution, right? If you're looking at a blood sample, there's tons of human DNA, and in that tons of human DNA, you want to find just a teeny tiny amount of viral DNA. So inhibition happens when Inhibition happens because like some of these, well, there's two issues here. Okay, let's start with inhibition. Inhibition means that like there's stuff in the blood that will actually inhibit PCR, the ability to amplify that DNA. All right. There might be other stuff that prevents the detection of that sequence of interest. So the first thing here is the concentration effect. When you partition, when you have very, very few amounts of molecules, let's say you have in a 16 microliter sample, you have just 2 molecules that you're trying to detect. So your concentration is, I should say 2 molecules, 1 molecule for every 8 microliters. That's a really tiny concentration. If you then partition the sample into many, many containers, what's going to happen, in this example, we've only partitioned it into 16 containers. But what we've done here, effectively, is that some of these partitions are completely empty, all right? So we're not interested in those. But in the partitions that actually contain a molecule, you've increased the effective concentration by 8 times. Because let's say you broke the 16 microliter sample up into 1 microliter partitions. The partitions that actually have a molecule, the concentration is now 1 molecule per microliter instead of 1 molecule per 8 microliters. All right, so that's a concentration effect that you're getting from digital assays. The second thing you're getting is an enrichment effect. This comes back to inhibitors. Let's say, in the case of nucleic acids, you're trying to detect small amounts of viral DNA or viral RNA. So that's shown in red here. And then simultaneously, but you have the soup of other DNA, like normal DNA, healthy DNA. That's shown in green. If you try to use PCR amplification to detect these small amounts of viral DNA, so an inhibitory effect that you're going to get is sometimes you'll end up amplifying all the normal DNA and not the specific sequences that you're looking for. All right, PCR is pretty good at detecting small amounts of sequences, but it's not perfect. Sometimes you end up with what's called unspecific amplification. So if you partition, you get this concentration/enrichment effect, where some of your partitions, the partitions that actually contain the sample, you may end up getting this this nucleic acid with none of the alleles or the wild type alleles on there. So you'll get a complete purification here. In other cases, you might have just 1 or 2. So in this case, you'll still get a large amount of purification. All right. So you're basically concentrating. You're getting concentration for free when you do partitioning. All right. So those are the reasons why you'd want to do digital assays. Now, what are the challenges? Well, one of the things is that which we haven't really talked about is, of course, you have to have a way of detecting single molecules in these partitions, right? So how do you actually do that? Fortunately, back in the 80s, there was a technique called PCR that was discovered. PCR is polymerase chain reaction. It's a way of amplifying DNA, making multiple copies of it. It may be very difficult for us to detect a single molecule of DNA, but if we first make many, many copies of it, then we can label it fluorescently with the fluorophore and then detect it. So that's for detecting nucleic acids. If you're trying to detect single proteins, you have to use enzymatic amplification. So I'm going to talk about a few examples of that over the next 10 to 15 minutes. So, this is one of the challenges. Second thing about digital assays is that you have to make sure that your sample concentration doesn't get too high. If your sample concentration gets too high, meaning like if there's an average of more than 10 molecules per partition, well, then you're not going to have any empty partitions anymore, right? If you don't have empty partitions, you're no longer in the digital domain. You're no longer in a digital assay. Then you're basically in this domain, the analog domain. All right. The partition volume should be uniform, and sometimes you deal with there's a surface chemistry in partitions. These partitions are really, really tiny. There's a high surface area to volume ratio, so sometimes the assays don't work quite as well as they do in the bulk. I think in the interest of time, I'm going to go through the mathematics quickly, so I'm not trying to pull a snow wash over all of you, but I just want to cover the applications. And if you're interested in the mathematics, I mentioned I wrote a review article last year, and I can refer you to that. We can talk further if anyone's interested in this. So really quickly, when you're doing a digital assay, what you're interested in is finding lambda. Lambda is the concentration, and it's a normalized concentration, so the units would be the number of molecules per partition, the average number of molecules per partition. All right, and that's going to be equal to m divided by n, where m is the number of molecules and n is the number of partitions. So when lambda is less than 5, typically less than 5 or 10 molecules per partition, digital assays can be used to back out what the concentration was. And this is the slide that I'm not going to spend too much time on, but I will just mention that Poisson statistics are very, very well established. They've been around for centuries. It relates the number of empty droplets to the sample concentration. I'm not going to go over the derivation here, but you start off with a binomial distribution, you simplify it to the Poisson distribution, and ultimately what you get is actually a very simple formula. Lambda is equal to negative ln of E. E is the proportion of empty partitions that you found in your assay. So if we finished our assay and we found that there are, like I said, like 33.6% empty and the rest are full. That 33.6% is what you plug into this equation here. Take the log of that and that gives you the sample concentration. It's very, very simple. And the mathematics are very well established, meaning that not only does it give you an answer, but it also gives you a confidence interval. I'm not showing that here, but you can say with okay, the answer, the sample concentration is this, with 95% certainty that it's within this range. It's very good for statistics as well. And that's one of the reasons why a lot of folks are looking at digital assays right now for diagnostics, right? Whether it's diagnosing, if you're trying to count the number of viral DNA that you have or the amount of cancer DNA that you may have in your blood and so on. So let's start with digital PCR. We may not get to all the digital assays today, but we'll definitely get through digital PCR. Digital PCR counts the nucleic acids of specific sequences. It has advantages over traditional PCR, meaning like you have a precise quantitation, you don't have to do a calibration curve, and it's able to detect really rare sequences because of the concentration and enrichment effect. On the other hand, the advantages of traditional PCR is that you get higher throughput and it costs less because you don't have to do this partitioning step. This is what a typical digital PCR assay will look like. There are several companies that have commercial instruments nowadays. Both of these, digital PCR relies on a chemistry that makes the droplets fluorescent when there are nucleic acids inside of it. So let's say you have one nucleic acid in your droplet, okay? What you do is that in the droplet they also put, there's also a PCR master mix and there's also an intercalating probe. So the intercalating probe is shown here. What that does is it's a special molecule that fluoresces when it's bound to DNA. If it's not bound to DNA, it will not fluoresce. So it's a way to detect DNA. Then there's these things called Tachman probes. I'm not going to go into the details of it, but it's a similar thing. It fluoresces in the presence of DNA. Now, the other part of this is PCR. And PCR is a technique that allows you to amplify the amount of DNA that you have. So even if you start off with a single molecule of DNA, at each PCR cycle you end up doubling the amount. So if you do like, let's say 40 replication cycles, you're going to end up with lots of DNA in that partition, and you'll be able to see it using fluorescence. So ultimately, in the end, we're not trying to measure the amount of fluorescence, we're just trying to detect whether there is fluorescence there or not. So a result of a digital assay might actually look something like this. You take an image, a fluorescent image, and some of your partitions become fluorescent. Some of your partitions are fluorescent, they light up. Those are the partitions that had DNA inside of it. So let's say one nucleic acid was in there, one or more, but usually it's just one. This particular diagram is actually showing multicolor, so it's actually possible to detect multiple sequences at once. Let's say you're not just interested in virus A, but you want to detect virus A, B, and C, or you want to detect 3 sequences that are known to be linked to virus A. so you can do multiple assays at the same time. All right, you see most of the partitions are empty, but some of them have nucleic acids inside of it. So that's what it looks like. So you'll basically count up the number of full partitions that you have, and you count up the number of empty ones. You get the proportion. You plug that into the Poisson equation. That tells you how many molecules of DNA you had in the solution. Very, very cool technique. There's two types of digital PCR systems that are on the market right now. And these have all come about just in the last like 5 to 7 years. One way, they're differentiated by the way they partition. One way of partitioning is by partitioning into arrays of microchambers. So Fluidigm out in San Francisco has a product, and Thermo Fisher has a product called Quant Studio. And both of these techniques, they take the sample, they take the sample containing DNA, they add the PCR mix, and they add the fluorescent probes, and then they partition it. Imagine taking a plastic tray that has a bunch of tiny little wells drilled into it, and you take your sample and you just squish it against that plate. Your sample is going to go into those wells. So it'll be partitioned into those wells, right? So that's one way of doing it, arrays of microchambers. Now they're called microchambers because they're super tiny. You can see here in this bottom left corner that each one of the wells here on the Thermo Fisher Quant Studio is only 60 microns wide. So these have to be made using microfabrication techniques, generally. All right, so these mechanisms all partition into an array of physical chambers. The second type of system, and these are actually, I would say, have done better commercially, uses microdroplets. And my lab actually does a lot of work with microdroplets, so that's how I got interested in this area. So this is where you take your sample again, your master mix, your fluorescent probes, and the way you partition is by using a microfluidic droplet generator. The way it works is that you take your sample coming in on the left here, and you mix it with two streams. You combine it with two streams of oil coming in from the top and the bottom. And when you mix oil and water together, they don't mix, right? They're immiscible. If you do that in a controlled microfluidic junction, you'll end up generating these droplets. And the droplets will be very uniform in size. So using microfluidic droplet generators, you can generate thousands of droplets, 1,000 droplets per second, and you generally only need, 20,000 to 100,000 droplets for doing digital PCR. So it doesn't take very long to generate the number of droplets that you need. So you'll first generate the droplets, and then you'll read. You have to do a read step. When you do reading, you want to find the fluorescence in each droplet. As I mentioned before, one way of doing that is by just lining up all the droplets in a single layer and then taking a picture of it. And you'll be able to see which droplets are fluorescent and which ones aren't. Another way of doing it, which is what the Bio-Rad system does, is that they flow all the droplets through something called a flow cytometer. And those of you who've done flow cytometry before, you may be familiar. Basically, each droplet goes through a focused laser beam, and when that focused laser beam hits it, the droplet either fluoresces or it doesn't. So you're doing this test you're doing one at a time. The droplets are flowing through the flow cytometer one at a time, and you're rapidly counting up the number of fluorescent versus non-fluorescent droplets. A flow cytometer will give you a chart that looks like this. This is actually a scatter plot of green fluorescence versus red fluorescence, and you can count up how many droplets you have that are fluorescent versus non-fluorescent. All right. But the analysis is still the same. You look at, you measure the proportion of non-fluorescent droplets, use Poisson statistics to figure out what the molecular concentration was. All right, applications. Since we have only 10 minutes left, it looks like we're not going to get into digital protein assays or their applications, but let's talk about a few applications of digital PCR since Caitlin and Kayla are working on viral diagnostics. So I think that you'll probably agree that one of the challenges in detecting viruses is that there's very little viral DNA compared to all this other stuff that's in the solution. So we've already talked about why digital PCR is, or why partitioning can help you because it isolates, it concentrates, and it enriches that viral DNA in each partition. So the applications of digital PCR are the applications that take advantage of what digital PCR is good at. It's good at isolating individual molecules. It's good at detecting very, very low concentrations of molecules down to the single nucleotide level. And so the applications can sort of be categorized into these areas. So rare mutant and allele detection, rare sequence detection, and then copy number variation. Copy number variation, let's just show you that real quick. This is one of the first papers published in Digital PCR was looking at copy number variation. Now, some of you may be familiar with trisomy, other types of mutations where you may, let's say you were supposed to have 2 copies of a gene, but because of some mutation you had either a deletion of that gene, you had an extra copy of that gene put into your genome for whatever reason. So being able to measure the number of copies of a certain DNA sequence that you may have, this is useful for human diagnostics. We'll get to viral diagnostics in a second. So one of the reasons why digital PCR is really good at measuring copy number variation is because of its precision. Because we're using Poisson statistics to actually count the number of sequences that we have, we can do that very precisely. Traditional PCR, quantitative PCR, can detect, I would say, a twofold change in copy number. So what does a twofold change mean? It means that you can detect the difference between 1 or 2 copies. You can detect the difference between 2 and 4 copies. You could detect the difference between 5 and 10 copies. A twofold difference. All right, but sometimes you may be interested in detecting copy number variation with much better resolution. Let's say you wanted to detect the difference between 6 and 7 copies. That's only a 10% difference. Digital PCR can resolve copy numbers, as you can see in this chart here. It can count 1, 2, 3, 4, 5, 6. It can tell the difference between copy numbers of all of these guys. So this is very useful for diagnostics in mammalian diseases that involve copy number variation. Now, quantifying viral load and then quantifying rare mutants, this is another area. So quantifying viral load, I mentioned there's very tiny amounts of viral DNA in your blood, and how do we detect that? Well, with traditional qPCR, it's difficult to quantify the amount of virus that you have because of these inhibitory effects. You end up amplifying other things instead of the DNA that you're interested in. So the low detection limits, in this case, it enables you to do early detection and to monitor the response to drug treatments. Sometimes we have viral DNA in our blood, and that virus is just getting started, right? When the virus starts to replicate, then the viral DNA will increase in our blood, and then we'll be able to detect it using conventional techniques. But what if we want to detect a virus early? It's very difficult to do that. Techniques like digital PCR allows us to detect viruses when they're in their nascent to early stages, and the earlier you can detect a virus, the quicker you can treat it. So for example, with HIV, if you have early diagnosis of HIV, you can treat it and suppress it much better than with later detection. Another thing is monitoring responses to drug treatments. Let's say you have a patient with HIV and you want to see how their viral load, the amount of viral DNA in their blood, how that viral load is changing over time with excuse me. You can use digital PCR to do that. The last thing I'll mention here is rare mutation detection. So this is a similar idea. You have circulating tumor DNA in your blood. Some of you may have heard about this area. If you haven't yet, this is one of the hot areas in biotech right now. It's called liquid biopsy. Traditional biopsies is where you go into the patient, you cut out the tumor, and then you culture the cancer cells, and you look at the DNA sequences on there, and you look to see like what's causing those cells to grow in an uncontrolled manner. With liquid biopsies, it's non-invasive. You're just doing a blood draw, and you're looking for stuff in the blood that's related to that cancer. Now, digital PCR is good at detecting circulating tumor DNA. If you look at this chart here, if you have a tumor somewhere, a tumor undergoes necrosis, or it gives off as the tumor is breaking apart, or I'm sorry, I guess as the tumor cells are growing and they're dying, they sometimes give off small amounts of DNA, all right? And so that DNA goes into the bloodstream. That DNA is almost identical to your normal DNA. And it's 99.99999% the same as your normal, but it's just that 0.0001% mutation that's in there. Sometimes it's just a single base mutation that causes that cell to become cancerous. So this is a situation where you're trying to detect DNA that's very similar to your native DNA, and so it's definitely going to be subject to those inhibitory effects. So digital PCR is good at detecting these really, really rare mutants in the presence of a large population of wild type alleles. All right, so this is an example of where they're able to detect cancerous cells versus non-cancerous ones using just detecting the tumor DNA in the bloodstream rather than doing a physical biopsy. All right, so I want to spend the last 5 minutes or so on questions. So I'll just mention that you can also there's also digital protein assays, digital cell assays, and so on, and there's very nice examples of that. If any of you are interested in working in this area, please do contact me, and we can chat. All right. So how about questions then? Yes, please.
So with that last technology you showed us, the ctDNA, are there benefits to looking at ctDNA over the CTCs themselves, like the cells circulating, tumor cells circulating?
Great question. The question was, is there an advantage to looking at circulating tumor DNA versus looking at the cells themselves? So there are, just in the last like 5 to 7 years, there have been numerous technologies that actually allow you to capture and isolate circulating tumor cells from blood. In fact, one of the companies is based here in Michigan. They're called CellSeek. Very, very, very cool stuff. It's a very hot area, but that will allow you to capturing the tumor cell will give you the cell, so you can look at cell phenotype. You can do analysis of. You can try treating that cell with various types of cancer drugs, and see if if any of them impact its growth. But if where this becomes useful, and this could actually be used on cells that have been captured already, it gives you information about the cancer's genomic sequence. There was a mutation in there, and those mutations are becoming better and better known right now. What causes a cell to become cancerous? They know what some of those mutations are. So this will actually enable you to amplify that sequence and determine what that mutation was, where that mutation happened. And the reason you want to know that is because cancer therapies now are actually becoming mutation specific now. If you know that the mutation happened in this particular codon or in this particular gene, then you probably want to use this particular chemotherapeutic agent. Other questions? Yes.
And so, in your example of the microfluidics and getting it down to the smaller partitions, it was just 1 microliter. But you had said, like, if you take 1 microliter and split it into multiple thousand droplets, how exactly does the microfluidics work in that way? Would it be similar to the droplet generation with the oil channels, or is that, or what is currently used? Yeah.
Actually, at the microfluidic domain, both approaches are actually used. One is you make a thing of small wells, really, really tiny wells, and you just put your sample on top of that and you squish it down. That's one way to do it. But the droplet approach, so our lab works a lot with droplets. The typical volume of these droplets is on the order of picoliters. That's how small they typically are. Much, much smaller than microliters. It would be very hard to pipette out 20,000 partitions. Historically, the first example of digital assays was actually done on a 96-well plate. They only had 96 partitions. But the advantage of using these microfluidic systems is that you can generate 20,000, 100,000, million partitions, and it turns out the more partitions you have, the more precise the assay is, the better the dynamic range is. So there is an advantage to using technologies to make more partitions.
In that type of microfluidic system, do you need to take into account small variations in properties of your assay that you're trying to do, or your sample? Like if you have a slightly different viscosity, is that a big deal? Like do you have to calibrate that or something?
Yeah, that's a really good point, especially if you're working with droplet type systems.
Right.
When you're generating droplets, the droplet size can actually be, it depends on a lot of factors, right? So typically, like if you buy a system like the Biorad system, they will have a very specific protocol of isolating your DNA, and then they'll give you the reagents for it, which have a known viscosity. So everything's been calibrated in advance.
All right. So how do you deal with that in a research setting?
In a research setting, so if I were to do this experiment, it's fairly easy to generate reproducible droplet size. You just don't know what that size is always beforehand. It'll be within a 20 or 30% bound, but it won't be several orders of magnitude off. But as long as the properties of the sample doesn't change, like you've made your sample, right? It has, and you've shaken it up, so it has uniform properties. Once you put that in the drop generator, all the droplets will generally be uniform size.
Okay. So it's more of a relative thing, and it only varies by 20 to 30%, so it's not such a big deal.
Yeah, it's not. But because you're doing some sample preparation beforehand, if you're trying to deal with raw samples and dirty samples where you just don't know where you're dealing with, then you're going to end up getting polydispersed droplets. And there are ways of dealing with that too. That's out of scope of this discussion.
So we're out of time today, but let's thank Dr. Basu.
All right, thank you. Thank you all. Yes, please.